We've now looked at different aspects of nucleotides and structural features of nucleotides, including the nitrogenous base, the phosphate groups, and also the orientation of the base, as it rotates around the glycosidic bond, and we've also looked at the puckering of the ribofuranose ring for these nucleotides. And while nucleotides do have functions by themselves, for instance, ATP, which is adenosine triphosphate, is itself a nucleotide. So that's involved, of course, as an energy currency for the cell, and it's also involved, or it's also a component of other energy molecules such as NADH or NADPH, right? But we also know that the point that we were trying to get at the beginning of this is what is going on with the structures of DNA and RNA. And so DNA, for instance, is going to be a polymer of the deoxynucleotides. And here's one of the deoxynucleotides in its general form. So the base is shown here as just a general nitrogenous base. And since this is the deoxy form, we don't have an OH group at the 2 position. And so the polymerization occurs then between the phosphate groups of 2 different nucleotides. So let's draw a second one here. So in other words, the polymer will look like this. Where we have the phosphate group, right? And this is going to be attached to a second nucleotide. Oops. Let's expand that a little bit more. There we go. All right, here's our next OH group. Okay, and then here's our base, okay? So, in other words, what the linkage between the monomers that eventually form the nucleic acid, which is what we call the polymerized form of this, is this phosphate linkage, okay? And so remember, we number these as 1 primed, 2 primed, 3 primed, 4 primed, 5 primed, okay? So the phosphate groups are at the 5 primed end. So the phosphate is connected to the 5 prime carbon. So we call this the 5 primed end of the nucleic acid. And then on the other side of the ribose, the other phosphate group is attached to the 3 primed end, the 3 prime carbon of this ribose ring. And so this is now then the 3 primed end. So you can think of this as, for instance, in a protein, remember we talk about the N terminal and the C terminal. So this would be analogous to the N terminal, whereas this would be analogous to the C terminal. All right? So by convention, we also write when we write a sequence, a DNA sequence, for instance, we know that DNA is double-stranded, but usually we only write 1 strand because the second strand is implied because these complement, right? So, but we write 1 strand, and we write it in the 5 prime to 3 prime direction. So the first nucleotide of this nucleic acid sequence is G, and then there's C, A, T, A, C, T, G, G, and G is at the 3 primed end of the sequence, okay? So the bond, or the functional group that links this has a special name. So this phosphate group that links the 5 prime nucleotide with the 3 prime nucleotide is called a phosphodiester bond. Okay, and again, that's that you can think of an analogy to, this would be analogous to a peptide bond. for a protein.